Hi, my name is Eric Niederkoffler. I'm with Thermo Fisher Scientific. I'm the R&D manager. And what I'm presenting today is a robust and sensitive workflow solution for qualitative and quantitative analysis of an antibody drug conjugate. So the challenges with an antibody drug conjugate is, one, the qualitative differences. For instance, you need to characterize it as far as the DAR ratio, as well as maybe the glycosylations that are associated with it. And then also you'd want to look at quantitating those. So the basic workflow here is we're using, shown in this figure, what we have is the Thermo Scientific MESA microcolumns, which in this case are coated with a streptavidin chemistry. We're going to use the Thermo Scientific Capture Select anti-IgG. That's going to allow us to now capture our ADC, which is based on a humanized monoclonal antibody, because this anti-IgG actually cross-reacts with those human IgGs. This is in a rodent plasma sample, so we're looking at preclinical studies. It can also be done in non-human primates. Using the MESA microcolumns, which are densely coated with the streptavidin, that are housed in a pipette tip, we can use different instrumentation to actually automate the process of doing the capture. So you can see here we have the TCAN multi-channel ARM96. We have the Thermo Scientific Versette, which allows you to process 96 samples as well. And then for a lower throughput, we have the Thermo Scientific Novus I and Stamp. So with these instruments equipped with the MESA microcolumns, we now can aspirate and dispense our solution containing the anti-IgG. So once that's loaded, it is now available to be used to capture our ADC, in this case from rodent plasma. After capturing the ADC from the rodent plasma, we then have a couple options. If we want to look at intact analysis, we can simply elute and look at the intact. If we want to look at a bottom-up approach, we can then elute out, do a digestion with trypsin, and look at the peptide map. So now I'm going to move over to some of the data that we've generated. So here we're looking at intact analysis of our ADC. You can see the chromatography. We use a monolithic column, which is the ProSwift RP4H column. You can see that this is very busy, and the reason why is the drug is conjugated through the lysine, so we have very multiple locations where it's been conjugated. And in fact, if we zoom in, and this is what is done here, we zoom in and we can see the different charges from plus 53 all the way up to like plus 56. And you see with the Q Exactive Plus series of instruments, we can actually get the resolution of each one of these. Very good instrument. We're actually only using the lower end of the resolving power of this instrument at 200 ml per Z. We're at a resolving power of 70,000, and it actually can go higher. So we can get more resolution on this if we wished. Taking this data, we can then use the Biopharma Finder software and actually deconvolute it. Now this gives you the ability to see all the payloads that are attached to this monoclonal antibody. And so we can see all the way up to a DAR 8 with this species, all the way down to a DAR 0, which has no drug conjugate on it. And in fact, we can also see where we have incidences right here, where you have the conjugation, but the payload actually is not on the ADC. So that's the intact. It gives you some qualitative analysis of that. I'm going to move to some studies where we started looking at that qualitative analysis and did a time course. So what's shown here in the time zero slots, which are the peaks that are facing down, is the ADC at time zero. If you take that ADC and spike it in rodent plasma, and we leave it out on the bench top for 2 hours, this is what's up on the top here. You can see some slight differences start to occur. The drug is actually starting to fall off. You can see kind of in the differences in the heights of these peaks from the zero. If we allow it to go for 24 hours, you can see we get a drastic change. In this case, we're seeing a lot of it come off, and now we're getting back to where we just have the monoclonal antibody and no payload on it. So again, we're able to use this technology to be able to look at the payload coming off and see the differences across it, any kind of transformations that occur, biotransformations that are occurring. So let's talk a little bit about the bottom-up approach. We've talked about intact, which gave us the ability to look at some of those differences. Now let's zoom in and do a bottom-up approach. We're going to digest it. And you can see here as we digest it, we get pretty good coverage. We're getting better than 79% both in the heavy and light chain in coverage of this ADC. A lot of the challenges could be with the fact that we have so many drug payloads on it. And then also, if we look at it, we can also see the glycosylation. We can see the sugars that are associated with the monoclonal antibody. So that's some of the results here that we're showing. Now that was all qualitative analysis. What about quantitative analysis? Because that's important when you're going to PK, PD, and ADME studies. And so we took a surrogate peptide that's unique to the ADC. I want to also note that when we go in a quantitative aspect, we actually have to have internal reference standard. So in this case, we used a monoclonal antibody, human monoclonal antibody that's available, and we spike it into the actual plasma samples along with the ADC that's there. And then the same protocol is followed to extract both that internal reference standard that we just spiked in and the ADC. And we use the ratio of the signal intensity of, in this case, this peptide from the ADC to a peptide from the internal reference standard, and that's what's being plotted here on the vertical axis. So we're actually looking at a ratio. What that means is through your entire protocol from sample prep all the way to data analysis, you're normalizing all the events that may occur. So for instance, if I'm running my ADC and I don't see the internal reference standard, then something went wrong. You can see we get a very linear dynamic range, ranging from 5 to 640 micrograms per ml. And then if we look at here, you can see kind of the results that we're getting as far as the controls. N of 5 replicates. You see the two different controls here, the concentrations we're looking at. You can see the CVs we're getting. We're getting better than 10% CVs on this. Accuracies, better than 15% accuracies across these two controls. So very good quantitative aspect and a full package workflow where it gives you qual-quant ability. Thank you for your attention.